Hey everyone, a massive welcome to all the 11th grade biology students tuning into today's explainer. Have you ever looked at a cut on your hand and just watched it heal over a few days? Or, maybe looked at an old photo and wondered how you grew from a tiny toddler into the person you are right now? Well, the fundamental mystery of how organisms grow, heal, and survive all comes down to a really remarkable, highly orchestrated process going on inside you right this very second. It's a story of preparation and division.
To really get this, we got to travel back in time and look at a famous philosophical quote from the scientist Rudolf Virchow. He stated, "Omni cellula est cellula." This simple little Latin phrase is actually the foundational hook for our entire understanding of biology and where cells come from. Okay, let's dive into this. Literally translated, Virchow's quote means, all cells arise from pre-existing cells. Sounds pretty simple, right? But think about the core puzzle here for a second. If every new cell is just an old cell that's split in half, how does it do that without losing all its essential parts? If you just rip a cell in half, eventually you'd end up with nothing. So how does a cell perfectly divide its incredibly complex internal machinery?
To solve this mystery, here's our roadmap for today. We're going to cover the origin of all cells, preparation versus actual division, G1, the first growth phase, the S phase, where we double the DNA, G2 and the M phase, and finally, G0, the quiescent stage.
All right, starting right off with Section 1, the origin of all cells, the absolute foundation of growth and reproduction. Moving right along to Section 2, preparation versus actual division, the 95-5 rule of the cell's life.
Now, what's really interesting here is a major misconception a lot of us have. When we think about cell division, we usually picture that dramatic moment of a cell tearing into two, right? But in a typical 24-hour human cell cycle, the cell spends a staggering 95% of its time. That's roughly 23 hours just preparing. We call this massive chunk of time interphase. The actual physical splitting, the M phase, takes only about 5% of the time. That's literally just a single hour out of the whole day.
So let's map out how this 24-hour journey actually breaks down. It happens in 4 specific steps. G1, which stands for the first gap, the S phase, or synthesis phase, G2, the second gap, and finally the M phase, mitosis, the actual division. Those first 3 steps, G1, S, and G2, are all grouped together as that massive 23-hour interface we just talked about.
Which brings us to Section 3, G1, the first growth phase, or as I like to call it, making the big commitment.
Think of G1 as the initial decision-making stage. The cell was just born, and now it has to decide, hey, am I ready to do this all over again? During G1, the cell is incredibly busy. It undergoes massive cytoplasmic growth, physically expanding in size, it's metabolically hyperactive, constantly making the proteins and enzymes required for growth, and interestingly, G1 is the most variable phase when it comes to time. Why? Because it's the only phase that depends heavily on internal genetic factors and external factors, like food and nutrients. If it doesn't have enough food, it's just going to linger there in G1. But listen to this. Once a cell finally clears that G1 checkpoint, it makes a massive commitment. It is locked in, for sure, and absolutely must complete the division process.
And that locked-in commitment takes us right into Section 4, S phase, doubling the DNA, the climax of the preparation phase.
Now, this is where the cell secures its genetic legacy. But I want you to pause for a second and just think about this logically. Why must a cell double its DNA before it divides? If it's already growing and getting bigger anyway, why does the genetic material itself need to be completely replicated?
So, the crucial point is this. DNA is the cell's ultimate instruction manual. You literally cannot build a brand new factory and only hand the manager half of the blueprints. If you just split the existing DNA in half, those 2 new daughter cells wouldn't have the complete instructions they need to survive. They need a perfect 100% complete copy.
Okay, so to make this happen, a few incredible things happen simultaneously during the S phase. Inside the nucleus, the DNA replicates perfectly. Out in the cytoplasm, if it's an animal cell, tiny structures called centrioles double up. You can think of these as little cellular fishing poles that'll pull the cell apart later. And finally, because DNA is incredibly long, the cell uses special packing proteins called histones to spool it up. Since we just doubled the DNA, we obviously have to double the histone proteins to pack all that newly synthesized material.
And this brilliantly illustrates one of the absolute trickiest concepts you'll face on your exams. The physical amount of DNA material, which we denote as C, doubles from 2C in G1 to 4C in the S and G2 phases. The physical material literally gets heavier. However, the chromosome number, denoted as N, stays exactly the same at 2N. How is that possible? Actually, think of it like a book with 46 chapters. In the S phase, you photocopy every single page and glue it right next to the original page. The book is twice as heavy now. The amount doubled to 4C, but you still only have a 46-chapter book. The number of chapters, the chromosomes, remains 2N until the cell actually splits.
All right, let's hit section 5, G2 and the M phase. Time for final checks and the actual split. So the DNA is perfectly copied. The preparation is ending and the real action is about to begin. The G2 phase is basically the cell's final pre-flight checklist. It's double-checking everything. It starts synthesizing specialized tubulin proteins, which form those spindle fibers needed to pull the cell apart. It's also the time when cell organelles, like mitochondria and plastids, which, by the way, have their own tiny bits of DNA, divide and double in number, ensuring both new cells will have enough cellular power plants to keep going.
Once that final checklist is fully cleared, we enter the M phase, mitosis. This is where the magic happens. And as we noted earlier, in a 24-hour cycle, this intense, highly choreographed splitting takes roughly just 1 single hour. This is literally the only time in the entire cycle where the DNA tightly condenses and the chromosomes actually become visible under a microscope. The cell divides its perfectly copied book of DNA, splits the cytoplasm, and boom, 2 identical daughter cells are born.
But wait, we have a plot twist. Let's check out section 6, G0, the quiescent stage, stepping off the ride.
So what happens to cells that decide to take a different path? Consider this. A newly born cell finishes the M phase and enters G1. But what if it's a super specialized cell, like an adult nerve cell or a heart muscle cell that has absolutely no need to replicate further? What happens to it then?
Well, it enters what we call the G0 phase, the quiescent stage. Now, we need to shatter a huge misconception right here. A cell in G0 is absolutely not a dead cell. It is metabolically active, highly functioning, doing its everyday job. It has simply stepped off the roller coaster of division. It completely exits the cell cycle. Some cells, like those nerve cells, stay there permanently, but others act as a reserve. If an injury occurs, they can jump right back out of G0, re-enter G1, and start dividing again to heal the wound.
Which brings us right back to where we started, with a final, provocative thought. If Virchow was right, and every cell comes from a pre-existing cell in this continuous, endless cycle of preparation and division, what actually limits the number of times a single cell lineage can divide before it just gets too old? That, my friends, is the secret of aging and cellular biology, and it's a fascinating topic for another explainer. I hope this really clarified the cell cycle for you all. Keep reviewing these concepts, trust the process, and best of luck on your upcoming biology exams.